and say, hello everyone, I'm Deb Paul, and I'm here with my fellow Darwin Core Hour team. They can all wave via the chat. And our guest presenters, Gabriella Drug, Katie Barker, and Andy Bentley. We're here to talk about sharing DNA and tissue data information using extensions via the GBIF IPT tool. On that, Gabby, take it away.
Okay, thanks, Deb. Final check. Do you hear me okay? Okay, great. Okay, welcome everybody. My name is Gabi Droege. I work at the Botanic Garden Botanical Museum in Berlin. And I work in the GGBN project for many years now. And I'm very thankful to the ITech Bio team for having us here as a guest session to tell you how to make your DNA and tissue collections available with the IPT. So, first of all, I can't expect that everybody knows GGBN well. So we will start with a brief introduction to GGBN and then also a brief introduction to the GGBN Data Standard. And then we will focus on how to use the GGBN Data Standard with IPT. I will guide you through the process with screenshots step by step. And after that, I will show you a final good-looking Darwin Core archive ready to be provided to GGBN. At the very end of this session, I will give you a short outlook of other things that we are planning related to the GGBN Data Standard, and then Andy Bentley will step in and tell you something about Specify and GGBN. And you will see that we do many things in GGBN, and I can't cover everything, but I will really try to select the most important pieces to give you an overview. And we invite you to come to our third conference in Vienna in May, where, well, it's a full conference dedicated to biobanking, biodiversity biobanking, organized by GGBN. So I will start with a problem. We have millions of sequences, as you know, and a couple of taxa, and not for all taxa exist sequences, of course, but the biggest issue here is that only for a few sequences there exist samples and voucher information. There might exist samples and vouchers in some collections, but mostly you don't know about it. So we want to help solving this problem and increase the number of vouchered sequences. Second, there is another big problem, which is the sample access. So the researchers need samples of high quality, and they should be well documented and, of course, legally obtained. And the reality so far is that the sample information is very fragmented. They are hard to find, and you have limited voucher information, and it's often ambitious sample quality. So this is another big problem that we want to address and solve. So, as you know, there is GBIF doing an excellent job taking care of specimen records. And on the other hand, you have the sequencing portals, AMBL, DDBJ, NCBI, and also BOL doing also an excellent job taking care of the sequences. But there is something in between the specimens and the sequences. And this is the physical DNA and tissue samples. And here is where GGBN steps in and wants to bridge the gap and wants to establish crosslinks between a physical DNA, a physical tissue, and its underlying specimen, and also the resulting sequences, publications, and so on. So the Global Genome Biodiversity Network is a global network of well-managed collections of genome quality DNA and tissue samples, but also environmental samples from across the tree of life, benefiting society through biodiversity research, development, and conservation. We are a network for non-human biobanks. So the majority of our members are natural history collections and botanic gardens. But we also do have culture collections and zoos as members. And in the near future, we want to reach out to other communities such as seed banks, viruses, domestic animals, crops, and ancient humans, because we all have to deal with molecular samples. And so we all share some of our problems, and we should find solutions together. Some of our members don't even have a repository. For instance, GBIF and ISPR are also members of GGBN, and of course, we appreciate their membership, so it's not necessary to have a repository to become a member of GGBN. GGBN was founded in 2011, and it had a precursor project called DNA Bank Network, which was founded in 2007 in Germany. And in 2007, I started working for this project. So I'm now involved for more than 11 years, working on the virtual infrastructure. The General Secretariat of GGBN is hosted by the Smithsonian Institution. So this is the job of Katie Barker, who is also with us today in the chat. She is the coordinator of GGBN. And the Technical Secretariat is hosted at my institution, the Botanic Garden Botanical Museum in Berlin. So I'm the technical manager. And the mission of GGBN is to foster collaborations among repositories of molecular biodiversity in order to ensure quality standards, improve best practices of any kind, secure interoperability and harmonize exchange of material in accordance with national and international legislation and conventions. Today we have 70 GGBN members worldwide. So it all started in Europe, but now you can see we are distributed across the world. We work on developing best practices in different areas. For instance, we work together with ISPR. ISPR is the International Society for Biological and Environmental Samples, and they focus on human biobanks. So they have a very good best practices document, and we helped to include special things about biodiversity biobanks to be included in the 4th edition. And this has been now published 2 weeks ago. And ISPR is also a member of GGBN. Second, we have the GGBN document library. It is a knowledge platform for non-human biobanking. It is open for everybody. And today we have more than 300 papers, reports, protocols, everything you can think of if you think of non-human biobanking. Of course, we only share documents where we have the permission to do so. And you can find it at library.ggbn.org. And this is a collaboration with ESBB. They also focus on human biobanking, but also on environmental samples. And another big topic is, of course, access and benefit sharing in the Nagoya Protocol. So in 2014, we created a set of documents that our members, but also everybody else, can use. Material transfer agreements for receiving or loaning material, the code of conduct, and a statement of use of genomic material. And this was a collaboration with CETA, which is the European Consortium of European Taxonomic Facilities. So our goal is to provide trusted and transparent access to genomic samples for users and contributors through an ABS framework. So the long-term goal is to have really a network of trusted collections, so to say, certified by GGBN if you're a member of GGBN, and that the exchange of samples between our members is as easy as possible under the legal circumstances. We also created an ABS FAQ that you can find in our wiki, and I'm sure Katie has already provided you the link in the chat. Another important tool that we have developed is the GGBN Data Standard. I will come back to this, of course, in a minute in more detail, but I want to mention it here already. It includes a permit vocabulary, which will be mandatory for our members from 2020. And we also want to improve the crosslinks between I and SDC, so this is NCBI, EMBL, and DDBJ, BOLD, GBIF, and GGBN. So we started a collaboration 1 year ago, and they were very positive about it, and we are now really working towards this goal. Another important thing about GGBN is that people can request samples through our data portal. And this request is then forwarded to the GGBN member holding the samples. The institution is then responsible for all further steps, meaning they have to check the availability of the samples, check again the compliance with ABS and their loaning conditions. Then they can offer a price or a service charge to the scientists and have to make sure that they get the signature of the material transfer agreement before they finally can ship out the samples. But requesting a sample doesn't mean that you really get the sample. It is the responsibility and the decision of the sample holding institution to give you the samples or not. And as you may know, if you loan and ship out samples, it's often hard to get a feedback on resulting publications and sequences. So we also work together with INSTC, BOLD, and GBIF towards a standardized citation guideline for the samples. This includes also a review of existing submission tools so that we can really give our members and the researchers a best practice how to do it today. And very important, GGBN does not own any samples. We provide the virtual infrastructure, but the ownership stays with the institutions, with our members. So to summarize this important topic a little bit, GGBN provides tools and best practices to deal with ABS and the Nagoya Protocol. And we also work together with other communities, initiatives, organizations, because our primary goal is to not reinvent the wheel if it's not needed, but primarily focus on gaps and try to fill them. So we provide a terms of reference, code of conduct. Our members must sign the Memorandum of Understanding, the ABS FAQ, the document library, the data standard, and finally the data portal. So in the portal, you need a set of mandatory fields to provide data, and it has a sample request system, and blocking of samples is also possible. So you can show them in the portal, but they are, for instance, blocked for loaning. So now, GGBN Data Standard. There is also a wiki page available. It's part of the TEDWIC wiki. There is the data standard documented. It is a set of terms for tissue and DNA samples, but some of the vocabularies are also useful for specimens, cultures, whatever you want to share. So for instance, the loan, the permit, and the preservation and preparation vocabulary. So they can be used for any kind of sample or specimen. The GGBN Data Standard is meant to be used with ABCD or Darwin Core. It is not a standalone solution. So this means in our data standard, all occurrence terms are excluded, geography, scientific name, etc. Because if you use it together with Darwin Core, we simply use the Darwin Core terms. If you want to dive in all details and background about the data standard, I recommend that you have a look at our white paper. You can also find it in our library, of course, or on our homepage under publications. We also, again, talk to other communities and to find other standards. So for instance, we included terms from the MIX standard. MIX is the minimum information about any sequence, developed by the Genomic Standards Consortium. And they have a lot of molecular terms already. And if they are useful for GGBN as well, there's no need to create new ones. We simply use them and just, again, fill just the gaps. So the standard is submitted to GSC and TEDWIC for ratification now. It is also supported by IPT since version 2.2 and today used by 5 GGBN providers. And the alternative software for providing data to GBIF and GGBN is BioCase, and there it is supported since version 3.5.3 and today used by 14 GGBN providers. So now comes the GGBN Data Portal. How does it all look like? So here is an example which highlights the most important thing about it, the relations. So in this case, you have a tissue, and it says it's deposited at Denver Botanic Gardens. And this tissue, if you would click on this tab, you would come to the specimen. So there is a relation between the tissue and the specimen record. And here, you see more tissue for this specimen. In this particular case, it is a typical population sampling often done by botanists. So you collect in the field one herbarium specimen and then 20 or 30 tissue samples from other individuals from the same population. And the portal knows that all of these tissues belong to this specimen. Here is another example, same case, but here you have DNA in addition. So again, it says there is more DNA for this particular specimen and more tissue. So they all are a big record family, so to say. So the documentation of genomic DNA samples in principle is challenging. Why? Because currently in our GGBN member repositories, it's just an estimate, but it's about 12 to 15 million samples that we have in our collections. And if I look further to the non-members, we might have 50 or 60 million samples somewhere in the freezers of this planet. So today only 6% are database and publicly available. And the legal aspects and the documentation are a major bottleneck, and the older the specimens, the worse. Also, the links to the voucher material is a major bottleneck. You know, there must be a voucher, but where? Maybe in a, you know, the particular collection, but you haven't recorded the particular voucher number, and the voucher specimen is not available in the database. So it's a big mess. So we have the GGBN Data Standard, and it is working well with ABCD and Darwin Core Archive. So what we now can focus on is trying to adapt the individual databases, which is now work in progress, but it will take some time because people and institutions are using different databases. Some don't even have a database for their specimens. And there is no alternative available. It might be that in a few years' time, we will find out that the whole model is working, but that there is a new great solution that how we could do it better. Fine with me, but currently the proposed way to do it is working very well. And so we hope that now all of our members can really adopt the system. So now comes the fun part, how to do it with IPT. Well, I guess you are very familiar with Darwin Core. That's the principal star schema. You have one core and a couple of extensions. And what GGBN does is simply creating a couple of further extensions. And a typical GGBN archive contains just a few of them, but always this Darwin Core resource relationship extension. So the important extensions, as I said, absolutely mandatory is the Darwin Core resource relationship, because here you declare the reference to your voucher specimen. Then the material sample extension, also mandatory, because here you declare the sample type. Is it a tissue, a DNA, environmental sample, a specimen, and whatever? And this is usually the thing that is not yet available in the databases, because usually you just say, I have leaf, muscle, whatever, but only a human knows that this is tissue. So we need to add another level, and this is covered by the material sample extension. Then the GGBN preparation extension, this is mandatory for DNA samples. Here you can provide the extraction method, date, whatever else you want to provide. It doesn't make sense for all kinds of tissue. For instance, if you just have collected leaf material, well, it's hard to tell how you did prepare it. But if you want to use it for a culture, it makes absolutely sense. How did you prepare this culture? What's the medium and all this stuff? And if you have NCBI or AMBL numbers or bold numbers, we recommend to use the amplification extension to share this information so that we can make sure the crosslinks to the sequencing portals are working. And the other two extensions that are very important, as I said earlier, it's the permit extension, will become mandatory from 2020. Here we cover things like, is a particular permit available or not? It has controlled vocabulary, or we have a big set of all kinds of permit types, collecting permit, export permits, MTA, MOU, whatever you can think of. And in 2020, the harvester will then check, for instance, the collection date versus the permit status. For example, if you have a sample collected after Nagoya came into force, and your permit status says unknown, meaning I don't know if I have a permit or not, well, this is unacceptable for us. If it's collected after Nagoya, you have to make sure you either have a permit or it was not required. And then you have to say why it was not required. But of course, we do not want you to share the permit itself. This is your internal documentation and not for the public. We just want to know, is it legally collected or not? And the other important extension is the loan extension. This is highly recommended because some people think it's not worth sharing information about, for instance, consumed samples. But I think it is important because they were used for research, that's why they are consumed. So probably publications or sequences or whatever referring to it. So make them available and just say it is available for loan. You can also block them because they are only used internally in your institution. If you block them completely, that they don't show up in GGBN, that's fine too. But this is something that you do on your database, not in the extension. You can also declare certain loaning conditions if you want. So it's a whole set of terms. So, of course, I don't want to give you an introduction to IPT. I expect you either that you know it already, or I recommend that you check out available IPT tutorials. So there are a whole series of great tutorials. So if you don't know IPT at all, please check them out. So if you are in your IPT, you go right into the administration, because you need to enable the extensions. So you click on core types and extensions. And here you see that all 3 core types, meaning event, taxon, and occurrence, are enabled already per default. So that's great. You don't have to worry about it. But the extensions, there are no extensions installed. So you have to do it. The IPT checks the GBIF vocabulary registry for available extensions. And a couple of years ago, we have already uploaded them to this vocabulary, so they are available for IPT. So you scroll down and you get a list. And please don't ask me why this is not ordered alphabetically. So it's a lot of fun to find the GGBN extensions. But then you simply click on the install button for the extensions that you want, and then they are installed. Again, we want to start with the resource relationship, material sample, and preparation as the most important extensions. So you start with creating a new resource like you would do for a normal GBIF mapping. You say a short name and you start with occurrence core. So we use the same occurrence core as GBIF does. Next, you can connect to a database, or you can select a file, CSV or TXT or whatever. I prefer database, so this is how it looks like. Just create an SQL statement and fine. And this is also a really nice preview tool where you can check how your data looks like when IPT tries to read it. Don't forget to save. And then you can continue with entering your metadata. And this is the metadata about your DNA and tissue collection, which might differ from the description that you would give about your specimen collection. But again, this is like a normal GBIF mapping, so technically just do like the IPT manual tells you. So provide the context and descriptions for your DNA and tissue collection and check the IPT manual. You must fill out the forms. As far as I know, there is no database collection for this metadata table, but I might be wrong. But it's not a big deal, just fill out the form, it's fine. So now we have the metadata, now we have a proper title, and we have the connection to the database and the metadata. So what we do now is start with the occurrence core mapping. Here you first, if you do an extension or a core mapping, the first thing you do is select a source. So it might be that you need to define multiple sources to enable the proper one-to-many relations within the IPT Darwin Core archive star schema. So you might have to create views or tables to flatten your database. All must refer to the occurrence ID. And I recommend to create different views for each extensions, especially if you have a one-to-many extension. So this is how it looks like, similar to the normal mapping. A difference here, collection code is something like DNA bank or DNA and tissue collection or whatever you want. And the occurrence ID is a GUID or whatever you want to provide. And of course, this is now the identifier for your DNA sample or for your tissue sample, not for the specimen. And in our case, I tested this with my own DNA bank. We provide stable identifiers for our DNA samples. So do a full GBIF-like mapping, including the catalog number, basis of record, and occurrence ID of your sample. For your samples, the basis of record always is material sample. So if you provide specimens to GBIF, you usually use preserved specimen or if you have cultures or the botanical garden, then it's living specimen. But here, we, together with GBIF, created a new term for the controlled vocabulary. It's called material sample. And you can use it for environmental samples, tissue samples, and DNA samples. And then just map the collecting event, scientific name, etc., like you would do for a GBIF mapping. There is a material sample ID field, which I think is no longer needed. So if you want to map it, provide the same value as for the occurrence ID, because originally there was a material sample core tested, but then we found out that the occurrence core is working perfectly for our purposes, so it was deprecated later. So you don't need the material sample ID, I think. So now we have the occurrence core mapping, and since we have installed the extensions earlier, they show up here. Next one is the material sample extension. A note appears which elements are mandatory. So it says it requires an ID, that's the occurrence ID, and the material sample type is required. Unfortunately, not all terms are displayed per default, so just click on Show All Classes, and then you will see all terms. But maybe it's fixed in the meanwhile. Don't know. I'm not checking IPT every day, of course. So here you see material sample is red, so it is absolutely mandatory, and you can either select a column from your table or just provide the literal. And here I just want to provide DNA samples, so I provide the literal. It's DNA and nothing else. And if you have questions about a particular term, you can click on the little icon, and then the description and examples show up and hopefully help you how to map it right. So again, why this material sample type? Because it's important to understand this. The basis of record is great, but it's top level. It's not precise enough. It just says material sample. Yeah, but what kind of material sample? So there is another Darwin Core term called preparations. This is also great, but it's too much free text. So here you would provide things like it's a muscle, it's leaf, it's a leg. Great, but imagine that I have 1 million GGBN records in the portal and you provide this free text. So how the hell should I know which one is a tissue and which not? I don't want to curate a big list of such free text. So the easiest thing to do is we need a simple classification. Is it a specimen, culture, tissue, DNA, environmental sample, to enable a proper search and filter feature. So in ABCD we have this term, it's called kind of unit. So for Darwin Core we created material sample type, which does finally the same job now. Maybe I should add, so here you just say it is a tissue, it is a DNA. And it's much easier for you to do it in your own database than I have to clean it on the top level, portal level. So the next extension is the preparation extension. And again, it says which are required, which terms are required as a preparation type and preparation date. So this is free text, but we recommend, if you provide DNA samples, that you use things like gDNA, eDNA, aDNA, and so on. And if you have a tissue or an environmental sample, you are really free to provide whatever works for you. We have made this available for search in the data portal, so people can search for this for the content here. But of course, it's much easier if it's some kind of easy vocabulary, so that people that can really find what they're looking for. And if you use a language different from English, for instance, it's of course a bit more tricky to find all liver samples, for instance. And the preparation materials, this is, for instance, the DNA extraction kit. Yeah, and if you made additional adaptations or additional steps in your extraction, then you can use the preparation process term, which is here. Okay, and now the most important extension, it's the relation to the underlying voucher. In the real world, you have, in this case, it's a little herbarium specimen, and you have a tissue sample. You might have collected both together in the field, and then you have a DNA sample. So you have specimen, sample, subsample. In the database, or better to say in the data standard, it's the other way around. It's the DNA sample having a parent record, the tissue record having a parent record. And why is it so? Well, if you imagine you have a population sampling, for instance, or whatever other complex sampling things, you quite often have one specimen and multiple tissues, and from each tissue probably multiple extractions or whatever. So you have a big set of samples, subsamples, and it's relatively easy to track all this in your database. But it's much more complicated to put all of this in a data standard like Darwin Core Archive or ABCD. So to make sure that the harvester and the portal at the end knows how to interpret the data, we came up with a solution that each record points to its parent and nothing else. And then the tissue doesn't care about the DNA, it just points to the specimen. And then on top of this sits the harvester, and it's asking the DNA sample, tell me your parent. And then it goes to the tissue and asks the tissue record, tell me your parent. And then it goes to the specimen record, tell me your parent. And the specimen record says, I don't have a parent, I'm fine. And then there are other DNA and tissue samples referring to the same specimen. So at the end, the harvester is aggregating all samples, all specimens that belong together. So to do so, we need proper identifiers, catalog number, collection code, institution code, occurrence ID for each of these records. So what you do is to provide it in the resource relationship extension. But there are no fields for catalog number, collection code, institution code. So what to do? The other thing is that the occurrence ID is not yet provided in a resolvable format by everyone. So many institutions now have a stable identifier. It's great. And if you click on it as a human, you might be referred to a specimen page on this institution, which is great. But as a machine, I can't read this data. There's nothing coming back. So we really hope that in the future we will have really linked open data, and that the machines can talk to each other and use these stable identifiers. So what we do now is we use a concatenated string instead. And it looks like this. So you provide catalog number is this number, and the collection code is botany, and institution code is US. So these are the identifiers of the specimen. So the tissue is referring to this specimen. And the specimen is available at this access point. So this is the Darwin Core Archive, in this case of the Smithsonian specimens. You can also provide the GUID or occurrence ID of this specimen. And that's all what the harvester needs to find this particular specimen at the end. And the harvester then goes to this archive asking for this record to get the full record details for this specimen. And in the future, as I said, we hopefully can use the stable identifier instead. So coming back to the portal, again, we have this relation, DNA referring to tissue, referring to specimen. And to do so, we have to use these extensions, and then it's working perfectly. This is also an example to show you how it looks like if you say sample cannot be loaned. Then there is this message here, and people cannot request the DNA. And it also says there is another tissue available for the same specimen. So keep in mind that sources and physical objects can be anywhere. I don't care, the portal don't care, as long as the data are provided properly. So here is an example. The DNA is deposited in Berlin Botanic Garden, and it has a relation to this specimen at the herbarium in El Salvador. So if I would click on this tab, it would show me this Lagu specimen, because my DNA database is connecting or referring directly to this external herbarium. And in this case, we have the permission to loan this DNA sample given by Lagu. In other cases, of course, we don't have. But this is also a very nice way, for instance, if researchers in your institutions are requesting specimens from other institutions to extract DNA. Then they get the specimen, you extract the DNA in your institution, but then what? You throw away the DNA. It's better to include it in your DNA bank too, but of course you are not allowed to loan it. But at least you can make it available, the information, and then the specimen holding institution can check the GGBN web service and then see where samples are still deposited. And that's a very nice feature, I think. Yeah, I'm almost done, Deb. If you want to find out more about mandatory and recommended fields for sharing data with GGBN, check out our website. You can also contact us directly at support@ggbn.org. Just a brief overview again: basis of record, material sample type, the preparation fields are mandatory, and again, the resource relationship. Permit status, as I said, controlled vocabulary, but this is all documented in the GGBN Data Standard page, so you can have a closer look there. This is how a final Darwin Core archive looks like. This is the Smithsonian archive, so you have a couple of extensions. I will quickly click through it, and Katie will provide you a link to the Smithsonian archive, so you can just download it and have a look yourself. So here is the material extension, material sample type. This is a fully mapped resource relationship extension with the concatenated relations. If you don't have a parent record, you don't have a row in this extension. The preparation extension and the permit extension. And here, this guy has 2 permit records for the same sample. Of course, you can provide as many as you want. Loan extension. And that's almost it. So the standard is now or currently implemented in Arctos and Specify. Andy will tell you a bit about it. We are also working on solutions for environmental DNA, environmental samples, and HTS library samples. You can check this out in our use case collection. There are examples available in our sandbox. So it's already working for ABCD and pretty soon also available for Darwin Core. And we're also planning a similar tutorial for the BioCase ABCD community. So that's it. Thank you, and I will hand over to Deb or Andy.
Hi, thank you very much for that information-packed presentation, Gabby. Wow. So if you stop sharing now, then Andy can jump in, and he's going to go quickly through this, so we have about 10 minutes left, I hope, at the end there for discussion. And I would like to say, please remember if you need to leave soon, to give yourself and us your present today. So please take that survey. And now could be a good time for you if you're needing to go or know you're going soon. And I hate to bother you with it, but it really is super important. So thank you very much for taking the 1 minute it will take to get through that. Andy, it looks like you're almost there. Yay. Can't hear you yet, though. Test. Might have to click your mic again at the top. Andy's sound was working before we started.
How's that?
Hold on.
Okay.
Now it's working. Yep.
All righty. Good morning, good afternoon, everybody. This is going to be a quick rundown of just some of the ways in which Specify has reacted to GGBN's portal and their data standard. There's two main things that we've done in Specify. We've amended our data model to include a number of the fields and tables that are required in order for this GGBN schema to work. And then we've also changed the way our publishing tool works. In Specify 6, there was only the facility to be able to run a single query, to be able to create an occurrence core. And now in Specify 7, we've amended that to be able to create all of the extensions through multiple queries and be able to provide all of that information. So looking at the data model itself, the Specify data model has been augmented. We had a number of the tables already existing in the system to be able to handle preparation information, DNA sequences and DNA sequence runs, the accessions, the permitting, the loan information, all of those kinds of things. Some of the changes that we've made, we've added a material sample table to be able to handle all of the information associated with the extractions and etc. We've added a primer table with a whole bunch of fields to allow the one-to-many relationships to DNA sequence runs and for you to be able to put in all of the information associated with those primers. And then we've added a number of fields to existing tables to accommodate a number of the schema elements. So this looks really scary, but don't worry about it. So the two new tables that we've added are there, the material sample table and the DNA primer table. And you can see that a bunch of new relationships have been created between those. So there is now a new relationship between material sample and the preparation table. So you can have, as a preparation, you can have a tissue, and then you can have a one-to-many relationship between that tissue and multiple extractions, which are then handled by the material sample table. And then you can have multiple DNA sequences associated with that extraction, and those DNA sequences can then be associated with multiple DNA primers. So once you get into your collection, you can then amend all of the forms, etc., to be able to accommodate all of this information. Here you can see a material sample table that includes all of the DNA sequence information down at the bottom there. We're not only handling regular Sanger sequencing, but we're also handling next-gen sequencing, so we have all of the fields to accommodate all of that kind of information if you need it. The next table is the DNA sequence run table, and you can see all of the fields associated with that, who ran it, when it was run, the primers that were used, etc., etc. All of that kind of information is available in those tables. And then you have the DNA primer table, which is in a one-to-many relationship to that, where you can keep all of the information associated with those primers so that you can keep more detailed information about the primers that are being used. So looking at the publishing side, the way Specify 6 worked is that we had a single occurrence core that you could create through creating a query through Specify that would then extract all of your data out of the database. So most of the mapping was done within Specify itself in order to be able to extract out that occurrence core. But you could only use a single query. You couldn't use multiple queries in order to be able to handle all of the Darwin Core extensions and get those working. You also had to have an IPT instance, whether it was yourself or somebody else's, in order to be able to then publish your data. So what we've now done through Specify 7 is created an XML process that you can go through in your collection to create multiple queries associated with the database to pull out an occurrence core and then all of the extension information associated with that. Obviously, one of those is GGBN, but there are others too. There's the Audubon Core extension for images, reference work, EOL reference work extension for reference works, and multiple other extensions that you can now use through Specify 7. We've done away with the need for an IPT client. You can actually publish it because Specify 7 is a web-based application. You can now publish your data directly through the web-generated Specify 7 instance and sort of circumvent the whole IPT process altogether. So what I'm going to do is just quickly show you what that looks like. So here is my ichthyology collection in Specify 7. If I go to my resources that are associated with that collection, you will see that there are a number of resources that are associated with the collection. If I go to my tissue collection, there are three things that are important here. One is the actual export feed itself, and we'll get to that later. And in the tissue collection, there is a Darwin Core tissue record and then a Darwin Core metadata record. That's all of the metadata that is associated with the collection. So if we click on the metadata record, you can see an XML file that goes through all of the metadata that you would regularly associate with an IPT client and produces all of those fields associated with the metadata of the collection itself. Then you create your actual occurrence core and extensions that go along with that. So here you can see all of the occurrence core information up at the top. And then if I scroll down towards the bottom, here is the GGBN extension. I haven't included the mandatory extensions as yet. We're still playing around with this and trying to get it sorted out the way that GGBN and Gabi need it. So I just stuck in a quick amplification extension to show you how it works. We also have Audubon Core extensions for collection objects and collecting events. And then we have a reference work extension down at the bottom. And so you can go through your collection, create a query that has all the necessary fields. You can then export that as an XML file, drop the XML file into here, add in all of the additional information, the terms and all of those kinds of things, and then set it up. And then your data will be extracted out using the occurrence core and all of those extensions associated with the occurrence core. Once you have those two files set up, you then go up and set up your export feed. And so this is the actual export channel that will then export all of that data out for both my voucher collection and my tissue collection as two separate zip files that then get sent out to the aggregators in order to be able to include my collections into the aggregators. So that's pretty much it. You know, we're still fiddling around with it and making sure that it works correctly. I'm playing around with it in the IDIG Bio sandbox and also in the GGBN sandbox to try and get it sorted out correctly. But once we have it all up and running and sorted out correctly, we can then start getting all of our Specify 7 clients using this instead of the old version. That's pretty much it.
Thanks very much, Andy, for that quick run through how Specify has integrated this, and understand that Arctos and others are working, and Symbiota as well, ways to share this kind of data. I would like to let Katie take over now. She's going to lead the discussion section. And I note that we'll be going over a bit on time today to get in this discussion. So please, though, if you need to go, don't feel bad. If you have a recording, you can come back for that. And if you have questions, get them in here before you go so that we can be sure to discuss them so you can hear the discussion around those, or contact us. And again, take that survey before you leave. Katie, take it away.
Thanks, Deb. I appreciate it. Okay, so the first question I saw was from John Vizoric, and it was asking about the amount of growth that GGBN expects and VertNet would expect by implementing the GGBN Data Standard. And I think it probably is highly correlated with the number of samples or institutions that are associated with tissue and DNA samples that would be publishing their sample information through these portals. John, do you have a ballpark idea of how many Arctos and VertNet providers there are?
Can you hear me?
Yes.
I don't know what the number is. I was hoping maybe Dusty could answer that question if he's still around.
Okay.
Yeah, but there won't be really new providers because I think almost all of them share their data through VertNet already. So it will be just another resource, data resource for each of them. So I think we were talking about 2 million or 2.5 million samples across the Arctos institutions, something like this.
Okay. And I believe the idea is that the VertNet is basically going to have the expanded vocabulary of the tissues and DNA fields, which allows them to share more information about their samples. Right, Gabby?
Yeah.
Yeah. I think it's the same idea with Specify. So, Andy, how many Specify users are out there? A lot, right?
Yeah, we have over 450 collections using Specify. How many of those have tissues, I don't know.
Yeah.
One of the things I was going to say is that there's potentially collections out there that are tissue only that may become providers now that they are able to, effectively map up all of their tissue information. I think Gabby's right. A number of the institutions are specimen holding institutions that have tissues as well, but there may be a couple of institutions that are tissue only holding institutions that may now be able to share their data. So you may see a slight bump in numbers.
Yeah. Also, we expect a huge increase of the number of samples available in the portal this year. So today we have approximately 400,000 samples plus 200,000 specimens available in GGBN. And for instance, Guelph will make part of its biobank available, and a couple of other institutions as well. So we're expecting more than 2 million new records during 2018.
All right. Are there any other questions about Specify and Arctos and GGBN?
I would add Symbiota too. I think Andy Miller would have known. Does anybody else from Symbiota here want to pipe up about latest developments? I'm not sure. I know that they have their changes to their software to make it
already.
So
they also try to implement it now.
Okay, so Symbiota has also tried to implement the GGBN Data Standard?
Not yet, but they have it on the list now.
I see. Good. Okay. All right. So Paula, I don't know if are you still there? Okay, cool.
Yes.
So you had a couple of questions about the occurrence core. Did Gabby and John answer that through the chat, or do you still have questions about the standard and the occurrence core? You may need to unmute yourself.
It looks like her connection dropped.
Oh, okay.
I'm not sure exactly. Do you guys see? I'm looking in the attendees, and I don't. Maybe she'll log back in. You guys,
No,
maybe you could clarify.
Still there.
I'm sorry.
She's as the host. She's typing into the chat.
Yes.
Oh, I see.
Yep.
Thank you. I had to scroll
There's
down.
the microphone.
Yeah, so maybe you guys can help. So it's still not clear to me about the metadata given that it would be an occurrence core.
Okay. We didn't want to create another core if we can use the existing one now. So if you imagine you have just a tissue, not a specimen, it's nothing else. It's connected somewhere by a certain date.
Gabby, you're fading out a little bit.
Oh, sorry. So a tissue sample is nothing else than an occurrence record. It was collected somewhere at a certain date by a certain person. So that's why we use the occurrence core, like you would do for an observation and a specimen. I hope this helps a bit. But you need to use a core. You can't use the extensions only to connect them to a core, and the occurrence core is the one that fits. Oh, there's a question for you, John. Does IPT run in SSL mode? I don't know.
I don't know the answer to that question either. I'd have to check that out.
Okay.
Maybe I can
Okay.
add something to what Gabby just said.
Yeah, Paula just said another question. Yeah, I think that's an IPT thing. So the metadata are just handled in this particular form. I think this is special to IPT. Maybe we can chat over email through this because it's really hard to understand what exactly you're asking for. You can use the same description for your DNA and tissue collection as for your specimen collection if you want, but mostly it is different. You can have a look in the portal. The metadata always appear below the specimen records, and just see what the others have done and compare it with the specimen metadata to see the differences.
Any other questions about the IPT?